Okay, so we'll kick off today's webinar. My name is Alison Quinn and I am the Marketing and Communications Manager for NIBRT. I'll be moderating today's webinar, so I'll talk you through how the webinar is going to run, when you have time for Q&As, how you can submit your questions, and I'll introduce our two speakers for today. So to begin with, I just want to give everybody a little bit of background information into NIBRT and what we actually do in NIBRT. So I'll just briefly introduce what we do. So NIBRT is a world-class training institute and research institute based in Dublin, whose mission is to deliver training and research solutions for the global biopharma manufacturing industry. We're based on the grounds of University College Dublin. We are actually 10 years in existence this year, so we opened in 2011. What we have in NIBRT that's quite unique is a state-of-the-art pilot plant facility, which is over 6,500 square meters, and that has all the equipment, all the technologies that are related to the biopharma manufacturing process. So trainees can come in almost like a flight simulator type process and go through all the processes and technologies involved in manufacturing biopharmaceuticals. We are established by the IDA in Ireland to promote world-class biopharma investment in Ireland. We are active on social media, so if you want to follow us, you can follow us on LinkedIn, Twitter, and also on YouTube. Just a little bit about what we actually do. So we support the biopharma industry by providing world-class research and training in our state-of-the-art facilities. So approximately 4,000 people are trained annually with NIBRT. We also collaborate with industry on a number of scientific research that drives biopharma innovation. We support the industry in Ireland. There has been major biopharma investment in Ireland, and the majority of these companies work with NIBRT to train their staff. We also provide a test bed for new technologies and processes. So if we look at what the biopharma sector in Ireland has achieved in the past number of years, we have over 75 biopharma companies in Ireland. You can see it's a massive and huge industry in Ireland. We have 10 billion in capital investment. It employs nearly 30,000 people in Ireland, and 15 of the top 20 biopharma companies have a basis in Ireland. We had over 73 billion of biopharma exports in 2018, and 50% of the top 20 products were manufactured in Ireland. So you can simply see how huge this industry is for Ireland and the importance of it in Ireland. We've won a number of awards for our facility, for our staff, for our innovation, and for our training. We also have won awards such as Partnership Alliance of the Year, Pharma and Education Training. We've had a number of our PIs listed on the Power List in the Medicine Maker. A number of the clients that we work with, so we work across the board with biopharma, with people that are in more contract manufacturing services, with pharma, with engineers, so across the board. So companies like Sanofi, BMS, 3M, Engineers Ireland, Shire, Wuxi, Amgen are all companies that we work with. So to stay in touch with us, please follow us on our website or follow us on our social media. You can also contact us with any queries that you might have at info@nibrt.ie. So to kick off the webinar today, today's webinar is being hosted with NIBRT and the Univercells Technology. The topic of the webinar is bridging the capacity gap for cell and gene therapies with technology innovation and skilled manufacturing workforce. Univercells and NIBRT have announced a collaboration to address the need for training in cell and gene therapies. So we're using next generation technologies for viral products manufacture. The immediate capacity ScaleX carbo systems from Univercells Technologies, featuring a fixed bed bioreactor and an inline concentration, which will be introduced in the Dublin facility for hands-on demonstration and training to support continued education and workforce sustainability. So we're very excited about this partnership. And today, John Milne, our Director of Training, and Mary Jordan, the VP of Marketing and Product Development and Product Management at Univercells Technologies, will be introducing our webinar. So to give a little bit of background on our two speakers, Mary Jordan is the VP of Marketing and Product Management at Univercells Technology. She oversees the company's product portfolio vision and development, as well as the marketing strategy and efforts to strengthen the company's market position and achieve desired business goals. Marie originally joined Univercells from the parent company in 2016, where she played a pivotal role in the launch of marketing and communications activities, including the successful commercial launch of Neverland as SIV PV platform and ScaleX hydro and carbo bioreactors. In 2020, she was a key driver in the creation of the spin-off company Univercells Technology. Prior to Univercells, her past assignments include project management at Baxter and research at MedImmune, now AstraZeneca. She holds a double master's degree in biotechnology engineering and in business management. So Marie will be delivering the second half of our presentation today, and there's plenty of opportunity to ask her any questions at the end of the session. We also want to welcome John Milne, our training director at NIBRT. So John joined NIBRT in 2013 in the role of training director, and his responsibilities include working with the training team who are responsible for delivering NIBRT training and education programs for both industrial and educational clients. Before he joined NIBRT, John worked as a consultant, working with several clients in the biologics area to develop purification processes and ancillary quality documentation. He graduated from UCD with a PhD in biochemistry, specializing in molecular enzymology. Following some additional academic research, he moved to industry, and he has over 25 years experience in the purification processes for proteins and peptide products. Within the biopharma industry, he has worked with Archport and Bioictrine, both contract manufacturing organizations. In his role as technical manager at Bioictrine, he was responsible for technical operations. Throughout his career in industry, he has performed a variety of roles for process development, scale-up, tech transfer, and GMP commercial production. So that's a brief introduction to our two speakers today. How it will run is that John is going to begin his presentation. His presentation will take approximately 20 to 25 minutes, and then we will pass it over to Marie, who will be approximately 25 minutes also. We'll then have 10 minutes at the end for any Q&As. So I'm just going to hand it over to John now to begin his presentation.
Thanks, Alison. You might just want to make me host there so I can just share a screen if that's okay.
Yeah. So you should be host now.
Okay, let me see. Can I do this?
Yeah.
Always the first hurdle, as they say. And hopefully people can see that now. I'll go into presentation mode. We should be good to go. Yeah.
Great.
So thanks very much, Alison, and thank you to everybody for dialing in on your Friday afternoons. I'm sure you're very busy people, lots to do. So thanks for taking time to listen to us today. And I'm pleased to co-present with Marie and highlight the partnership that we've just commenced with respect to NIBRT training. I'll probably be a little bit briefer than Alison suggested, because I'd like to leave more time actually for Marie to go through some of the real technical capabilities of the technology. I won't go over the NIBRT piece because Alison has done a good job on that. And I suppose the two things I really wanted to talk about today really was just to set the scene, perhaps for those people that are on the audience who are not as overtly familiar with advanced therapies. And then I'd also talk a little bit about the skills agenda, which is obviously close to my heart and NIBRT. And, you know, there's some tremendous products and tremendous innovations coming in our industry. But at the end of the day, the products have to be made. And for now, at least, we still need people heavily involved in that process. So I'd like to talk a little bit about the skills agenda. And if you look at, you know, people can look at surveys and literature and conferences. We know what's going on in this bold world of biomanufacturing. It's a tremendous landscape at the moment. There's a lot of good things happening, even within our existing industry, which to date has been dominated by monoclonal antibodies. And that's going to continue for the next period of time. That's not changing. And you'll hear people talking about diversification of pipelines among companies, multi-product manufacturing, flexibility, agility, increased use of single use, the increased interest in digitalization, so-called Industry 4.0, what that might mean, and then perhaps even embracing more intensification of manufacturing, leading to continuous manufacturing strategies. And there's a lot of exciting work being done in that space. But the other area that is clearly garnering a lot of attention is in the area of personalized medicine, most notably around the area of cell and gene therapies. And there's no doubt that there's a tremendous future impact from ATMPs. They offer groundbreaking opportunities for the treatment of very vexative diseases. And we tend most often to categorize these therapies into what we call cell therapies, gene therapies, and maybe even tissue engineered products. And I'm sure people are familiar, at least in concept, with what that actually means. But from a sort of a general perspective, advanced therapies start with either donor patient cells and or recombinant gene technology. And there's different ways for this to take place. And there's multiple companies across the world working in different aspects of this developing industry. We've already seen our first approvals. And there's no doubt, listening to regulators talking, that the number of approvals is likely to increase substantially. And that puts a lot of pressure, obviously, on the manufacturability of these products and some of the technical challenges that these therapies will present. And Mary, in her presentation, will talk a little bit around that, focusing on the gene therapy viral vector space. But again, just to give some information to people who maybe aren't as familiar with the concept, I think most people are probably more familiar with the term gene therapy and the idea that, we can replace a gene. As everybody knows, genes provide the information for the production of proteins in our body that are crucial to maintain life. And if, for example, you're unfortunate enough to be born with lacking a gene, so you don't have that protein, we're now embracing ways, rather than, for example, replacing that protein that somebody doesn't have on a sort of a continuous basis. Is there a potential that we could almost reboot the computer? We could give people the ability to be able to make that protein product. And this is what gene therapy really sets out to do. So we can replace the gene, for example, that causes a problem with a healthy copy. We can add genes to treat or fight disease, and perhaps even turn off genes or silence genes that are causing disease. And this can be done in an in vivo setting where you generate the vehicle, if you like, to introduce the transgene, and it's an injection into the patient. Or we can take cells from that patient and we can modify those cells, what we call ex vivo gene editing, take those cells, expand them, and reinfuse them back into the patient to carry out the activity. So again, we talk about in vivo and ex vivo, two sides to our business. When we start talking about cell therapy, and it's interesting because we've been using cells, we've been using stem cells for many years. We've been using bone marrow transplants in patients for many years. But when we talk about personalization of medicine and what this industry is really looking at is the potential that we can use what we call gene modified cell therapy, where you're using both a cell and a gene therapy modified outside the body to be then infused back into the patient. And a good example of this is what maybe some people have heard of, what we call CAR T cells, where you can harvest T cells from a patient. You can reprogram those cells outside of the body with a specific gene that's delivered by a viral vector. Those cells are then expanded, and those cells will then will elicit on their surface a receptor that is then directed against the specific cancer that the patient is suffering from. And we have approvals in this space already. And we have a lot of companies actually who are looking at potentially other cells. So not even a T cell, but looking at other cells in the immune system that could be amenable to this type of therapy. What I'd like to talk a little bit about is, a lot of people often ask me, is the learning curve, is there such a, what does this landscape look like with respect to manufacture? And what I want to really try and give people a sense of is that if people can manufacture biologics, if they can manufacture recombinant proteins and monoclonal antibodies, there's a tremendous degree of overlap. We are not necessarily dealing with apples and oranges here. And even like I just did a quick screen grab of a typical mAb process and a typical viral vector manufacturing process. And you'll notice the same terminology, same types of flow, same types of equipment. So again, there is a transferable skill set. The one advantage that monoclonal antibodies have, I suppose, is that they have probably 15 to 20 years of manufacturing experience under GMP conditions at large scale. So while they're not necessarily generic platforms, there's a high degree of platform aspect to what we do, and most companies set out to purify their own particular antibody, for example, in pretty much the same way, in the same strategy. So they have benefited from that, a degree of experience. And you'll hear people talking about gene therapy, for example, and they say, well, gene therapy is probably where antibodies was maybe 15 years ago, and that's probably a fair assumption. So over time, as vendors get more into this space, as more approvals come, we will probably find that things will become a little bit more templated in time. And this was a nice figure, actually, I got. If people haven't seen this document, it was put out by the Bioforum Operations Group, or BPOG, late last year, and they have a series of roadmaps and manufacturing maps. And I just for today, I just took one that relates to AAV production. And again, if people are familiar with traditional, what I call traditional biologics at this stage, like mAb production, you'll begin to see that in the process stages, there's a degree of overlap. We talk about the same things. We talk about cell banks. We talk about seed expansion, cell expansion. We talk about fermentation. We talk about purification. We talk about filling. Like, it's all there. The tools of our trade are there. You could argue that the piece here, stage 2, which is around manufacturing transfection solutions, that's different, but again, there's experience. People who've manipulated E. coli cells for traditional biologics have experience in this space. So that's probably an area where there's a nuanced difference, but it's not insurmountable. We've experienced that. So the point really being is that we shouldn't be afraid or scared by this. AAV production, as I like to say, is an evolution. It's not a revolution. The same requirements are required for people. People have to have skills, they have to have training, they have to have the correct attitude. And I often find in a NIBRT setting, when we're engaging with trainees, the attitudinal side of training is often the most challenging, okay? Because it's about a mindset, it's about doing the right thing all the time, when it really matters. But if you look at the sort of Venn diagram there, and we talk about recombinant protein, we talk about gene therapy, cell therapy, there are core technical disciplines that are common to all three. And if you spin that out into the operational techniques, the unit operations, cell culture, chromatography, filtration, viral reduction, impact of single-use systems, the whole notion of contamination control, aseptic technique, environmental monitoring, all of that infrastructure is well tried and well tested. And I'll show you a little bit later in this presentation one or two references to what the regulators say about ATMPs with respect to training of individuals. So it's not as if we're reinventing the wheel completely here. We're going to leverage off as much of the industry as what we have, and then add to it as that may be required. And one of the initiatives that NIBRT in Dublin has been interested in over the last couple of years was bringing a group of stakeholders within our own country together, from research, from academia, from government, from industry, to try and understand from our perspective what the skills and education and training framework might look like. And we went out and we interviewed various people. To build up a picture of what this is. And again, this is just for emphasis to show that in particular areas of advanced therapies, there were certain recommended skills and priorities that people were identifying with us. Two areas, for example, that people said that there was a clear need was in the area of analytics, characterization of some of these entities. And that's something that constantly came up in conversation. Also, maybe the wider approach to regulations, because one of the issues tends to be that when companies are bringing their product, for example, or their therapy to the regulators, it's almost at this stage like it's case by case. There isn't a tremendous amount of standardization yet, because it's still very early in its infancy. And when we talked to companies about cell therapy, they were calling out very heavily the practical programs in cell therapy operations, around cell isolation, cell expansion, cell engineering. And one obvious thing that struck me as somebody who's worked in the industry is, when you're making a monoclonal antibody, you're moving through your cell culture, your purification. You have a large volume of material. It's filled, it's formulated, it goes to the patient. You're treating multiple patients, and you tend not to think so much about that in those terms. When you're working in, for example, a cell therapy facility, you will have somebody working at a cabinet, for example, who may very well and probably will be working on cells for one specific patient. So really, you're getting into very, the skills piece around that, the knowledge that people have of what they're doing. It's a mind shift, it's a paradigm shift in terms of that, because you're literally making the product very often for the patient of one, particularly if you're generating an autologous therapy, which is the patient cells being treated and being developed and being moved back into the patient at some point. The other piece that was called out in our work in this framework was around the move towards digital skills, and that's happening in our industry anyway. You'll hear people talk about Industry 4.0, but it is likely that digital skill priorities will be central to the overall success of cell and gene therapies going forward. And there will be priorities and skills required around data modeling, real-time monitoring for technology for process and operational control, predictive analytics, machine learning. The whole supply chain around personalized medicines is very different to what we currently have. Very often you make a cell therapy, you may have 48 hours maximum to get that to the patient in another country. So it's presenting challenges. And if you're taking cells from a patient in a hospital and you're moving them to another facility to be developed in some way and then move back, you have all those supply chain logistics. So I wouldn't like you to think that there aren't challenges. There are challenges, but there are also opportunities, and this is what the industry will embrace. So just for the last piece, I want to talk a little bit about was around the best place I always find to look for information is when you look at what the regulators are asking. And the EMA in late 2017 put out their guidelines on GMP for advanced therapy products. And if you read that document and you read the very standard accepted ICH-Q7, for example, which is about GMP manufacture, and you isolate various pieces. So I was thinking primarily today about personnel and what does that mean? And you begin to see things that the regulators are asking for. And I mean, if you were to sit down and try and write this document yourself with a piece of paper and a pen, you probably would get very close to some of the points that have been raised in the document. You need adequate people with appropriate qualifications. They have to be experienced. An interesting comment was that all personnel involved in manufacturing or testing of ATMPs should have a clear understanding of their tasks, including knowledge of the product appropriate to those tasks. You should have, all your personnel should have training on the principles of GMP. Again, not surprising in a GMP environment. There should be periodic training. It should be assessed. You should monitor the effectiveness of your training programs. And particularly when people are working in aseptic environments, they need specific nuanced training, including in the aspects of contamination control and microbiology and frontline gowning. Again, nothing surprising. But again, if people are not familiar with the regulations, if you read that document, it gives you a very good sense of what's required. And then that brings you on to the question of, well, how do people get those skills? And as Alison said in her introduction, we've been training people for 10 years, and we like to think we do a very good job at that, because we think we understand what's best required for training individuals. And the experts on learning psychology will tell you that you probably only remember 10% of what you read, which is actually quite low when you think about it. But you remember 90% of what you do. So learning by doing, or what we call active engaged learning, is really what it's about. So, and that stimulates higher order thinking, problem solving, troubleshooting, and being able to critique and analyze. And that's at the heart of our programs in NIBRT, actually, for that reason. And when we go out to industry, which we do every year, we do a survey every year with the Medicine Maker in London, and we ask a series of questions. And we ask, I think our most recent survey, I'm showing you some information here, we had about 120 people, 125 people on the survey across the world at different levels and different size facilities. And what was called out was on-the-job training or practical training in a lab or a pilot plant environment as being the most intrinsically valuable things. And why is that? Because it's the doing. It's the active, it's engaging and troubleshooting. And I think that's a lesson. It's a nice comment for us, for our board, because it shows the value that people put on and want to spend dollars in our training facility. So what I would say in summary is that for competency-based learning to be effective, you must have trainees that engage with best-in-class equipment, using best-in-class training sessions, getting trainees to engage, in meaningfully and to think about what they're doing. And again, the BPOG group in 2016 put out a nice white paper on training for biotech operations. And if you haven't read it, there's a reference there to it. And it actually takes you on a sort of a journey as to how you build that skill set. And there's no doubt that that type of approach is still directly transferable to new emerging therapies that we will see. And the interesting thing about all of this, learn by doing, is that it's not a new concept at all. I mean, there's been great minds have thought of a long time ago about this, and what we have to learn to do, we learn by doing. And it's as true today as it was, nearly 2,000 years ago. So in summary for me, before I pass over to Marie, who's going to talk about the technical challenges and how they can be overcome, what I would say is that ATMPs will continue now to play an increasing role in therapeutic regimes and positive patient outcomes. The challenge of ensuring the personnel are qualified will always be an important challenge. We are a heavily regulated industry that is not going to change. Your people are always under scrutiny when it comes to audits. Competency-based programs will definitely equate with success, and that companies who genuinely go beyond just training people for compliance. What I like to talk about is training people for work. So go beyond what's the regulatory requirements. We'll find that your staff are more educated, they're more motivated, they'll develop those critical skills, and that has to be a win for both the employer, the employee, and most importantly in all of this, the patients. So I think that's me for now. I'm happy to answer any questions at the end of the webinar, or if people want to follow up with me by email after at any point about NIBRT or what our model might be, please do. And I'll pass over to Marie to take on and talk a little bit more about the technicalities of our partnership.
Great. Okay, I'll just pass over to Marie now. And okay. So Marie should be the host now. You should be able to pull up your slides.
Yes. So good afternoon, everyone, and thank you for joining. I am Marie Jourdan, the VP of Marketing and Product Management at Univercells Technologies. And during this second half of the webinar, I will be discussing how a combination of technology innovation and upskilling manufacturing workforce can contribute to bridge the capacity gap for viral vector manufacturing for cell and gene therapies. My talk of today will be structured as follows, with first a brief introduction of who we are. Then I will talk you through some of the specificities and challenges of gene therapy manufacturing and how these direct some choices when it comes to technology selection. And lastly, some initiatives that are already being taken to help upskill the teams in academia and industry to build the expertise of tomorrow to deliver on these gene therapies. So at Univercells Technologies, we was incorporated last year as an affiliate of the Univercells Group with the aim to design and deliver next evolution of biomanufacturing technologies. By doing that, we aim to support gene therapy developers and vaccine developers to advance their portfolio and deliver on high capacity while reducing their manufacturing costs. And this is done via an innovative technology approach, which combines intensification and training principles, which I will detail in just a moment. And today we are extremely proud to collaborate with Nyberg, which is for us a renowned global center of excellence for research and training, and that shares the same ambition to help advance the biopharmaceutical manufacturing industry. And we are particularly interested in their holistic approach of learning that John just detailed, that combines theoretical learning and hands-on experience. So just a couple of words of introduction on Univercells Technologies' approach to tackle virus manufacturing that combines the two principles of intensification and chaining. So first of all, our core technology strongly relies on the principle of intensification, first of all, starting with the fixed bed bioreactor. And how that works is that we have alternate layers of a fixed bed matrix where cell can adhere, and a spacer layer where the media can flow through the different layers and the cells. And these alternative layers are structured in a spiral wound manner, as detailed here. That really enables to offer a high growth surface for the cells to grow in a very drastically reduced volume. And in that structured design, the biomass is thus immobilized in the matrix that and delivers a high cell density for cell growth. And with this novel design, this really enables homogeneous cell distribution in the fixed bed and homogeneous media flow and nutrients availability to the cells. Also promoting a low shear stress environment for the cells. Altogether, we have noticed that this really helps increase the specific productivity of the cells and delivering higher raw throughput. And secondly, by design, this intensified fixed bed bioreactor is truly scalable by design by keeping constant the physical parameter across all different scales. So next to this first principle, we implement a chaining approach, which means that we link additional unit operation, such as here illustrated a TFF. Since the cells are trapped in the fixed bed, media can be perfused through the bioreactor, and that enables here continuous concentration of the raw product and harvest in a dedicated system. And that really delivers pre-clarified concentrated bulk products. And additional unit operation can be integrated from cell culture to concentration, clarification, and UFDF. And with these two approaches, we really aim to increase the raw performance and at the same time reduce the footprint of the operation, the volume usage, and thus enables to deploy equipment in much lower footprint, reducing your facility size and thus capital expenditure, and at the same time increasing the performance and thus reducing the operational cost of your process. And these two technology principles have been deployed in our portfolio of equipment that is comprised of Scalex bioreactors and a Neverland upstream platform. So we can see that there is, we start with a small scale bioreactor, a Scalex Hydro, that provides 2.4 square meter of surface available for cell growth. That's particularly suited for early feasibility and initial process optimization. Then in the middle, we have the uniquely positioned Scalex Cabo system that offers 10 and 30 square meter for growth surface, and that is suitable for R&D, process optimization, but also for GMP production, for instance, for clinical or commercial manufacturing, depending on the scale. And lastly, the last large-scale system is the Neverland upstream platform that incorporates the Scalex Nitro bioreactor, offering up to 600 square meter of growth surface, but that is also combined with inline clarification and concentration of our products. So combining upstream and midstream operation in an equipment of 3 square meter footprint. So that can be deployed in very low footprint facilities while delivering a high capacity for viral vectors. Following that, I will now give some illustration of some gene therapy specificities and manufacturing challenges and how that dictates your choice of technology. As John already highlighted, the gene therapy market is quite new and fast growing. We see that there are now hundreds of gene therapy that are heading to commercialization in the next few years. One specificity of these markets and of these therapies is a strong heterogeneity. That's illustrated here in this graph. We can see the dose sizes and the prevalence of some indication, and that they can vary drastically with up to 7 logs of differences in those sizes. So that brings first complexity when it comes to defining processes and selecting your commercial scale capability. And as John already mentioned as well, there is a strong gap in the manufacturing workforce to help design and deliver on these therapies, with some estimation of a need to double the workforce in a 5-year time frame. So what are then now the main goals, the main challenges of gene therapy developers to be able to bring their portfolio to the market? The first obvious need is the need to deliver on capacity. to deliver on the appropriate dose size to reach the target patient that they have. And for that, selecting the appropriate process and technology. There is also an evident need for speed for the gene therapy developers to be the first to reach the market and to reach a significant market share. And lastly, need to grow the specific skills and manpower that will be required in the very next months and years. So first of all, looking at what criteria needs to be taken into account to help select the appropriate technologies for our vector manufacturing. First of all, the first important consideration is anticipating your target scale, depending on your dose size and indication. Also, some low indication therapies, the plastic flatware technologies such as cell factories are still the technology of choice and can deliver sufficient capacity, reliability that one may need. Even though it is a scale-out approach, it can be perfectly suited. But as we see for gene therapies, the dose sizes can be much higher. And so for medium and higher demand, the developers have switched to more scalable approaches. And then there is a second criteria that needs to be taken into account, which is relying on your expression system and other process parameters, but mainly depending on if your cell line is in adherence mode or in suspension mode, this will really dictate the choice of technology. So historically, gene therapies have relied mainly on adherent cells, such as in the vaccine industry. And for that, fixed bed and packed beds bioreactors that have been introduced in the market a couple of years ago are well suited, increasing the VAR productivity in a compact footprint to deliver on higher capacities. But they still brought some limitation or constraints. And so some developers are now switching to suspension mode cell lines in order to be able to use more well-known stirred-tank bioreactors. So these are also a technology of choice to deliver on medium to high capabilities. However, they can face some limitation and especially still require a large footprint, large capabilities when it comes to stirred-tank bioreactors of 2,000 to 5,000 liters. So depending on your facility constraints, they might also not be the technology of choice. And we've introduced recently a new platform, which is the Neverland platform, combining an intensified fixed bed bioreactor with inline midstream processing that is suited for both suspension and adherence and line. And that by combining additional integration and increasing the VAR productivity can also offer a viable solution for high demand by keeping a very compact system and thus low footprint for your facility. So this is a first illustration of the technologies that can be suited for your commercial goal. Another consideration is how to bring your current research portfolio to this commercial capacity. So looking more specifically at the scale-up approach in suspension stirred-tank bioreactor and how the novel intensified continuous fixed beds compared to this more traditional approach. So of course, when scaling up, the aim is to maintain constant some of the key parameters to ensure that the cell environment remains the same and that you obtain similar results. So one of the first parameters to keep in mind is the gas exchange that allows homogeneous cell respiration and stripping of CO2. This is characterized in stirred-tank and in fixed bed bioreactors in the same manner, looking at your KLA. A second parameter is looking at your mixing condition that ensure appropriate nutrients availability and temperature distribution, and that can also still maintain low shear and low pressure on your cells. In the stirred-tank bioreactor, this is characterized by the power number, and the equivalent parameter that one need to monitor in the fixed bed is the linear speed of the media through the fixed bed. And then one last important parameter that you can take into account when looking at the different technologies in front of you is the geometric coherence of your bioreactor. And this will really influence the ease to scale up or to apply scaled-down models in your system. In certain bioreactors, you will be looking at some of the key dimensional ratios. And in fixed bed bioreactor, the main parameters that enable this linear scalability are the volume surface ratio and the fixed bed that needs to maintain similar compaction and similar height throughout the different scales that will take you through your commercial manufacturing. Now, as an illustration, I will expose just a short case study on NIV manufacturing. This project aimed to do a rapid technology evaluation comparing 2D to these 3D bioreactors, aiming to demonstrate equivalent performance between the two systems and highlight the potential for further process optimization. These case study targets were first to maintain the productivity, cell growth, and bar productivity between the two systems after the tech transfer. Secondly, to already initiate some reduction in material costs through the reduction in PI and DNA usage and media usage. And lastly, also optimize the seed train size as another way to optimize the cost of goods of the process. As a start, there was an initial technology transfer of the process from cell factories to fixed bed bioreactor. And how that translates is as follows. We can see here the high-level upstream and midstream layout of this process, which is fairly simple. First step of inoculation, obviously, followed by a growth in the bioreactor in recirculation mode. Transfection was achieved in batch mode, followed by VAR production, also in recirculation mode. And in one of the last experiments of this project, the cell lysis was followed by inline TFF for VAR's concentration and bioreactor rinsing to further improve the VAR product recovery in the end. And next to this initial tech transfer, some other process optimization were introduced, such as lowering the pH set point to reduce the usage of base in the process, a reduction of the inoculation density as a way to reduce the transfection mix that is also quite costly in VAR vector manufacturing, and lastly, reduction of the volume surface ratio in the growth phase as a way to reduce the usage of media. And here are some of the results of this first case study. First, looking at the cell growth profile on the left, we can see that we reach a similar high density in the bioreactor compared to the reference process, and up to 14-fold increase in cell density at the day of harvest. And we noticed that bioreactor 3 and 4 had a lower seeding density, but followed the same growth profile than the first two. And then on the right, looking at the resulting VAR productivity, the first bioreactor was a simple technology transfer with no optimization of the process and reached already 45% of the productivity compared to the reference process, which is in the expected range in that situation. And then the different optimizations were implemented from bioreactor 2. And we can see that in the fourth bioreactor, where we also implemented inline concentration and bioreactor rinsing, we could also reach 98% of the productivity of the reference process. So this initial set of experiments really aim to demonstrate the ease of tech transfer and reproducibility between 2D and a 3D bioreactor, and also the potential for further optimization. So here in only 4 bioreactors, we reached similar performance and initiated some cost optimization. And this can all, of course, be pursued in subsequent process optimization projects in that bioreactor. So this first set of experiment was done at small scale. And here is an extrapolation of the performance that would be achieved at commercial scale in the Neverland platform. So here with the a 600 square meter bioreactor that will be combined with inane concentration. And what we can notice is, first of all, by using this intensified and integrated process, first, this that drastically reduced the footprint of the unit operation and of the clean room that's required. And that has a tremendous impact on the facility CapEx. And then, secondly, through increased VAR performance and decrease of material usage, this also enables here up to 6-fold reduction in the cost of goods of the raw products. So now that I've discussed a bit how some process parameters and capacity constraints can help you select the different technologies that will be suited for you, I will start discussing a bit more about the need for the upskilling the teams to deliver on these new modalities, and how technology innovators and centers like Nyberg can collaborate to help growing the different teams for that applications. So first of all, one first way to foster knowledge growing is through smart equipment design and some selection criteria that can already help with the adoption of new technologies. So first of all, when designing and when selecting a technology, it is essential to ensure that this can be rapidly assessed by the Genotherapy innovators. So all that can be achieved is by enabling a simple deployment in a lab environment that can also ensure that novel technologies can rapidly be transferred from a reference process, as I demonstrated in the previous case study, and altogether that you can easily demonstrate the equipment performance and gauge if yes or no this technology is suitable for you. And that really aims to alleviate the workload of technology assessment for you and your team. And a second important aspect to keep in mind when designing technologies is to help ensure fast learning curve and ease of use of these new modalities. So as John also explained, there's a lot of transferable skills from the workflows that already works on similar modalities, whether in vaccines or in mAb. So ensuring that these team can rapidly understand and adopt novel technologies for gene therapies. And one new aspect is, as also John mentioned, a higher level of automation in these advanced therapies, heading toward digitalization that do require a new set of skills, but that will eventually help simplify the operation and upskilling of the workforce and de-risking your manufacturing. As an example of that, here are some of the more concrete parameters that are similar in 2D to 3D. So all the cell principles, cell inoculation, growth, infection, and VAR production remain the same, whereas in 2D flatware and in fixed bed bioreactors. But some new features comprise, for instance, additional parameters that need to be monitored in such fixed bed bioreactor and automated systems. For instance, we have a lot of sensors now monitoring the DO, pH, temperature, biomass, for instance, that the teams need to be familiar with, but that often come in an automated mode that enables a feedback loop and automatic adjustment of the parameters. And secondly, these systems often help to limit the manual operations. So for instance, in the Scalex Carbo, we offer pre-assembled single-use manifold. The tubings are guided on the backsheet or on the hardware to facilitate the installation, to avoid mistakes. And basically, the only remaining manual operations are the sampling of the liquids and of the fixed beds at some specific points through the process. So that really helps, for instance, some operator being able to operate different systems in parallel and removing the risk of manual errors. And lastly, one last feature is the availability of predefined recipes that enable the manufacturers to define their process configuration, their process parameters in a very flexible way, offering different options from fed batch perfusion, for instance, or infection and transfection. And then once this recipe has been defined for the process, the entire process is automated and runs smoothly through the control system. And that really also helps improve and maintain batch-to-batch reproducibility. So all these new features and considerations do require specific trainings for smooth adoption of the technologies, and eventually will help facilitate day-to-day operations and increase the reproducibility and reliability of the gene therapy processes. And one way, and this is why we have partnered with NIBRT today, is upon the launch of their new gene therapy course that is being designed. So during their course, this will help the trainees to discover in more detail the key consideration for viral vectors manufacture, deep diving even more in the unit operation, the process parameters that apply for these applications, and eventually enabling you to define your gene therapy parameters and to apply your knowledge on the latest technologies. So since this is combined with hands-on demo and training on a range of equipment. And at Univercells Technologies, we decided to feature the Scalix Carbo system because we believe it illustrates some of the key innovation that specifically address the gene therapy needs in terms of intensification, change in line operation, of course, automation and availability of predefined recipes that the workforce needs to get familiar with, and some other features like fixed bed sampling for direct cell monitoring. And this also provides a scaled-down model of the Neverland platform, the large scale, and so can already introduce some of the concepts regarding scalability and bridging the gap between R&D and commercial manufacturing. This training is being designed and will be available in NIBRT's catalog from September. So I invite you to pay attention and contact them if you're interested. And I thank you for your attention, and I welcome any question that you might have.
Great, thank you very much, Marie. So anybody who has a question to ask, please feel free to pop it into the Q&A box, or you can also pop it into the chat box. We have a question already, so it's for John, I think, really. It's more towards Nyberg. So the first question is, will Nyberg be introducing more Atmp-focused courses in the future?
Yeah, thanks, Alison, and thanks for the question. Yes, indeed, we are. So one of our aims coming into 2021 actually was to look at our curriculum, and in response to some inquiries we were getting, just I should say as well, Ireland has just recently welcomed the first new manufacturer into Ireland. Myra GTX have come in to manufacture viral vectors and make plasmids on site as well. So it's beginning to turn in an Irish context as well. So we've been having some conversations with those folks. But as in parallel, we have about, I think, 7 or 8 courses that are in development. One we've piloted already, and these courses will typically be 3 to 4 days, a blended approach, but spending time in our practical suites using some of the technology, including what the Scalix Carbo that Marie has just identified. And just as an aside to that as well, we are also in the process, slightly more longer term, but in the next 12 months of, but we are closing out a design phase for an expansion of the Nyberg plant, actually, which is going to embrace cell and gene therapies across research and training. So we'll be getting new training suites and putting in their equipment that really is in its own context. So that's something exciting for the future. So yes, we will. And if you keep an eye, as Alison said, on our website or our media outlets, you'll see notes about that, and there'll be more details as to what those courses will look like. We will be issuing a new catalogue, I believe, Alice, in the next couple of weeks, yeah, and in that there'll be some other information on it as well. So
yeah, all of our new course dates will be updated in the next couple of weeks, so we'll have a new brochure with all the dates for the rest of the year.
Yeah,
great. Okay, and the next question is for Marie. On what application has the system been tested?
Thank Thank you for the question. So the Scalix system has been tested on a range of applications. So in terms of adherent cell lines, the usual, we've started with Vero cell lines. We also have tested MRC5, HEC293, and some other cells like EV66. And we also recently demonstrated suitability in suspensions modalities with, I think, CHO cell line and HEC293 in suspension as well. So that really demonstrates the potentiality of the platform to be used independently on the choice of process. And in terms of our vectors, talking about gene therapy, I think the usual ones, lentivirus, AV, adeno, and then a range of our vaccines as well. And then some of the modalities such as proteins.
Okay, great. Thanks, Marie. John, how has NIBRT, through its offerings, planning to provide training solutions in the cell gene therapy space, and what might the timelines involved be?
Yeah, thanks, Alison. It's actually a little bit related to the first question, which is good. It's people are interested in training. So, I mean, our approach, first of all, is, as I said, we are, as an entity, we are expanding our facility. That will take 12 months, but that's something that's very active, and that will give us a lot more capability. And we have just started to reach out to the vendor community to understand the likes of partnerships that vendors may be willing to enter into with us. But in the short term, it is we're involved in several conversations with the higher education institutes in Ireland, because a lot of the universities and colleges and technical universities are looking to, I suppose, improve their curricula and start at least embracing new therapies in their programs. And we may see some specialized courses coming in the next year or so in that area. And NIBRT will have a mandate, I suppose, to do our best to support those programs. So it's really to tool up, to build our curriculum, and then make sure that we can offer that to as broad an audience as we can. So that's what's driving one of our, that's top of our mind with respect to advanced therapies. But we always have to be mindful that there's a big bad industry, bold industry out there as well that we also need to look after. So this is why we require, an expansion to our capability to give us that extra capability to be able to embrace more training. But it's a very active plan for NIBRT over the next, from now on, really, from the next year or so.
Right. Okay. And Marie, just one more question. What is the GMP track record so far?
Yes, good question. So, so far, both the Scalix Carbo and the Neverland upstream with the Scalix Nitro are designed to be suited for GMP manufacturing. And so far, most of our clients are in early stage and early clinical phase. And we've heard recently that one of the customers did successfully deliver and release a batch that's meant for clinical phase 1 material and testing. So that is the first success, and we know that many other customers are now entering the clinical pipeline as well.
Great. Okay, well, we're coming up to the 2 o'clock mark, or the 3 o'clock mark. So I think we'll finish the webinar off for today. If anyone has any other questions, please submit them to info@nybrт.ie, and I'll make sure that they're passed on to John and Marie. We can get your questions answered. So thank you very much, John, and thank you, Marie, for that excellent webinar. And the webinar will be available on demand for anyone who would like to have a copy of the recording.
Thanks, everybody. Thanks,
okay,
Alison.
Thanks,
bye-bye.
Everyone.
Have a good weekend. Bye-bye.
Bye. Bye.